Hello everyone, and welcome to today's The Scientist webinar. I'm Betsy Young, Interim Director of Creative Services for The Scientist, and I'll be moderating our discussion. Today, we'll be hearing from Leena Gandhi at NYU on just how far we've come with immunotherapies for lung cancer, and why immunotherapies represent a brave new world in which previously untreatable diseases are getting a much-needed second look. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the panelists will address these during the Q&A session following the presentation. To ask a question, simply type your query into the question box located on the bottom left-hand side of your screen. We will try to address as many of these questions as we can during our Q&A session. I apologize, it sounds like someone listening has their speakers on. We may need to ask them to turn them off. I apologize, it sounds like someone listening has their... The webinar platform is user-friendly. You can move or resize any of the windows by simply grabbing them at the top or stretching them at the bottom right-hand corner. You may need to move or minimize some of the windows to see the live view. The webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'm wondering if I'm able to ask for the other participants to mute their lines. We're getting some feedback and background noise. I'd like to take this opportunity to thank our webinar sponsor. Beckman Coulter develops, manufactures, and markets products that simplify, automate, and innovate complex biomedical testing. For over 80 years, our products have been making a difference in people's lives by improving the productivity of medical professionals and scientists, supplying critical information for improving patient health, and delivering trusted solutions for research and discovery. Scientists use Beckman Coulter life science research instruments to study complex biological problems, including causes of disease and potential new therapies or drugs. From basic cellular analysis to advanced cell sorting methods and flow cytometry applications, count on Beckman Coulter for efficient and reliable solutions to today's research challenges. Scientists can also optimize their workflow and increase laboratory efficiency with our liquid handling, centrifugation, and custom integration solutions. I think the feedback might be coming from our own scientist team. Would it be possible to mute the line? Thank you. And with that, let me introduce today's speaker, Dr. Leena Gandhi. Dr. Gandhi completed her PhD in molecular and cell biology at the University of California, Berkeley. She went on to get an MD at the New York University School of Medicine. She completed residency training at Massachusetts General Hospital and fellowship training in hematology-oncology at Dana-Farber Cancer Institute in Boston. Her research focuses on early drug development in lung cancer, and she led the clinical trials program in thoracic oncology at Dana-Farber. There, she worked in the Early Drug Development Center prior to moving to NYU Perlmutter Cancer Center last year. She currently serves as Director of Thoracic Medical Oncology and specifically works on developing novel immunotherapy combinations and understanding biomarkers of response and resistance in immunotherapeutic combinations. Dr. Gandhi?
Thank you. I will just go ahead and start. I'm really going to be talking primarily about immunotherapy as it pertains to non-small cell lung cancer, but I think it serves as a model for the development of newer drugs in this space in all solid tumors. I won't really be talking today about the development and the progress of immunotherapies in hematologic malignancies, although we might touch on that at the end, as I think that's sort of a whole webinar in and of itself. I think in order to understand what kind of an impact immunotherapy has had in lung cancer, it's helpful to sort of start by understanding the scope of the problem in lung cancer. Lung cancer is the #1 cancer killer in the U.S. There are over 200,000 people who were diagnosed in 2016, with the majority of them dying. That's unlike the most common lung cancers overall, breast and prostate cancer, which do not cause nearly the number of deaths that lung cancer does. The reason that lung cancer is such a cancer killer because more than 75% of patients are diagnosed with an advanced disease that is not curable with surgery alone, and more than half are diagnosed with metastatic disease at presentation. 47% of those new diagnoses now happen in women, and that percentage is increasing. Where we have been for many years in lung cancer, and the reason it's been such a difficult problem to treat, is that the natural history of lung cancer is often very aggressive. This is a Kaplan-Meier curve. On the bottom curve, you see what the natural history would be with best supportive care and no treatment at all, where patients lived for a median period of time of less than a year, close to 7 months. With the addition of chemotherapy, that was extended minimally, only by about 2 months. And that's really been our standard treatment paradigm for many, many years for the majority of patients. And although that slide was now a long time ago, many, many different attempts at trying to improve upon a particular chemotherapy regimen yielded basically absolutely zero difference whatsoever in terms of actually improving patients' survival. And you can see here from using 4 different platinum-based chemotherapy regimens, 2-year survival sort of remained stuck at around 11% overall. So there was really nowhere to move but up after that. And the one area that we have moved forward, and for a lot of patients who have oncogene-driven cancers, is in the area of targeted therapies, which I will not talk about today. But even in that realm, for those subsets of patients who get really significant benefit from targeting a specific driver mutation in cancer, those have not resulted in cures or long-term survivals. They have resulted in significantly improved response rates and significantly improved progression-free survivals, which basically shifts that survival curve over to the right. But it really, ultimately, all of those patients end up at the same place, which is at the bottom of that curve, with very, very few patients living for more than a few years. Immunotherapy in general is not a new concept in the treatment of cancer. It's something that people have been trying to do for many, many years. And intrinsically, it would make sense that if we could harness the power of your immune system to target cancer cells, which we know are abnormal, we know have acquired changes compared to normal cells, that would be a potentially very effective way to treat cancer. The problem is that for most cancers, most solid tumors, there's very little immune cells actually even within and around the tumor. Renal cell carcinoma and melanoma are unique in that they tend to have high levels of immune infiltrate within the tumor itself. And it was because of that that initial attempts at immunotherapy, which mostly consisted of cytokine stimulation, such as using IL-12, were done in these 2 tumor types. And what you can see here is that although these survival curves look reminiscent of those lung cancer curves, and that many people initially die within the first 1 or 2 years, there's a flattening of the curve, or a tail of the curve, for both melanoma and renal cell carcinoma, where a small number of patients had long-term survival. The problem is that it was very difficult to predict who those patients would be, and they were so few that it really has not become a standard of care. IL-12 or interferon, which is also used in melanoma, are drugs which really do not work for the vast majority of patients. And it still, again, leaves a lot to be desired in terms of effective treatment. So I would put IL-12 and interferon into a category of immunotherapy that I would call active immunotherapy, or trying to stimulate a response against tumors. And those could include cytokine-based therapies, but that also include cancer vaccines, which have also been an area of active interest for many years in many different tumor types, and one that has really not been really successful. In other words, there's not been dramatic differences in long-term outcomes with the use of tumor-specific vaccines or tumor antigen-specific vaccines. Passive immunotherapy, on the other hand, which is really the opposite, blocking inhibition at the tumor or at the tumor microenvironment level against the immune response, is a newer concept in oncology treatment and really got its start with the development of anti-CTLA-4 antibodies, and now more recently has transformed the field with the development of anti-PD-1 or PD-L1 antibodies. So I'm sorry, I think these are out of order. I'm just going to skip ahead to this slide, which sort of shows both, I think, active immunotherapies as well as passive immunotherapies are really working to modulate T-cell activity, since T-cells are really the worker cells of the immune system that are actually able to kill cancer cells. And we know that T-cell activation occurs through first recognition of an abnormal antigen, an abnormal peptide presented by an MHC complex, which you can see diagrammed at the top here. And that recognition that occurs via the T-cell receptor on the T-cell can lead to either activation or downregulation or suppression of the immune response, depending on other cofactors as well as the nature of the peptide interaction. So the signal 2 in the middle of this diagram here shows you the interaction of CTLA-4, which is a co-stimulatory factor for T-cell recognition that occurs specifically in CD4 T regulatory cells and enhances a population of suppressor cells or regulatory cells that are actually often found within tumors. And we see this actually quite frequently in melanomas. We see it much less commonly in lung cancers, but they're seen sporadically in many different tumor types. Anti-CTLA-4 antibody-based therapy was first evaluated in melanoma because of that prevalence of T-cell infiltrate, many of which were T regulatory cells or FOXP3 positive cells, and was actually a successful monotherapy and has been an approved therapy for melanoma for now several years. The difficulty with anti-CTLA-4 antibody-based therapy, the approved agent is Ipilimumab or Yervoy from Bristol-Myers Squibb, is that it, again, works for a subset of patients, although it's a higher subset certainly than IL-12 or interferon, and is associated with pretty significant autoimmune toxicities by changing the regulatory T-cell phenotype and percentage. You really do expose patients to potential significantly increased risk of autoimmune problems. Signal 3 at the bottom of the page details sort of the PD-1 and PD-L1 or PD-L2 interaction. PD-1 or programmed death 1 is a receptor on T cells that really serves to downregulate or suppress T cell activation. It happens when PD-L1, the ligand for the PD-1 receptor, or PD-L2 binds to PD-1 to shut off T cell activation, actually leads to programmed cell death. We know that PD-L1 and 2 can be expressed on dendritic cells and other immune cells, but also on tumor cells. And specifically PD-L1, which has been much more extensively studied, although PD-L2 expression seems to mirror that for in large part of PD-L1, that it can be highly expressed on some tumor types and some tumors. I should say not tumor types because it's not universal. It really depends on the individual tumor. In melanoma, PD-L1 expression is relatively high, on the order of many of about 40% or more expressing relatively high levels of PD-L1. In non-small cell lung cancer, it's a little bit different. There's a whole range of expression levels from 0 to 100%, and it really seems to be quite variable depending on other tumor characteristics, which we'll get to momentarily. But this sort of just diagrams a little bit more specifically that PD-1 inhibition that can happen when you target this interaction with either an anti-PD-1 antibody or an anti-PD-L1 antibody. So the PD-1 receptor on the T cells serves to inactivate T cells when it is bound by PD-L1, which can come from either tumor cell suppression or other immune cell suppression to modulate the immune response. we think this is really one of the key ways that tumor cells can evade the immune response, is really by locally suppressing T cells adjacent to the tumor and in an adjacent tumor microenvironment. Both PD-1 blockade, which there are several now approved agents in that class, including durvalumab, pembrolizumab, but also nivolumab and atezolizumab. I'm sorry, let me just clarify. So nivolumab, pembrolizumab are both PD-1 antibodies which are approved. There are now also multiple PD-L1 antibodies that are approved in different tumor types, which include durvalumab, avelumab, and atezolizumab. And in lung cancer, the first of these to be tested and the first to be approved was nivolumab, which you can see here from this study, which was a phase 1 study that had a large lung cancer cohort within it, that there are, excuse me for a minute, the screen for me is very small, so I'm just going to look at my own slides so I can read to you what's on there. But you can see in the top left panel the overall survival of patients and this similar tail of the curve that we saw with IL-12 and melanoma and renal cell, but we didn't see it all with chemotherapies and lung cancer, is happening for patients with these drugs, that we're seeing some patients, close to 20% actually, who are long-term survivors with immunotherapy. And these are patients with metastatic disease who otherwise, in most scenarios, would often not live longer than a year. The panel on the top right is actually just looking at different dosages, but again, you can see that the dose that went forward and is now an approved dose, 3 milligrams per kilogram, at 3 years, there's a 27% survival, which is, again, unheard of in lung cancer, where I told you that at 2 years, there were really only 11% survivors. At 3 years, almost 0. So for these patients, and it is still a subset, they are really getting long-term benefit, something that we really don't see with chemotherapy, and we don't really see with many targeted therapies either. You can see in the bottom left the spider plot, which shows you the responses that don't happen necessarily right away and continue to improve over time, so that they actually, sometimes you get the deepest response even up to more than a year after the treatment has initiated. One thing I will kind of summarize here in this next slide is a really ongoing controversy about the predictive value of PD-L1 expression on tumor cells in terms of being able to generate this response. We know that only a subset of patients are getting this long-term benefit, but it is really still an open question of how best to identify those patients. Some trials suggested a higher response and better long-term outcomes in those with high levels of PD-L1 expression, and this was really most clearly demonstrated with Pembrolizumab, where Merck, who makes Pembrolizumab, actually looked at an entire range of expression and came up with 50% PD-L1 expression not by intensity, but by the proportion of tumor cells expressing PD-L1 at any level as a cutoff to suggest that there was a higher chance of benefit versus a much lower chance of benefit if you express anything less than that, or 0%. PD-L1 expression overall happens in about 25 to 70% of non-small cell lung cancer, and that range is really because how you define PD-L1 expression is variable from study to study, from company to company. Every company that has developed each of those drugs I mentioned to you has a different assay, a different antibody that they use, as well as a different assay system for the antibody. So their definitions of what they call positive really vary from study to study. In addition, most of the other agents that have been studied looked at much lower cutoff levels, either between 1% or 5% or at most 25%. All of those ranges, if you looked in the Merck studies of Pembrolizumab, behaved very similarly. So those other studies that only looked in those low ranges didn't really find a difference with PD-L1 expression. Merck did, maybe because they were looking at a much higher level. The other problem in identifying what is the true expression level of PD-L1 is that it's a dynamic biomarker, and I'll come back to this point a little later, but that really can affect your interpretation depending on when you get the biopsy, where you get the biopsy, and how you get the biopsy. this slide just shows you actually how Merck defined their interpretation of high positivity and how this led to differential use of the drugs in subsets of patients. But you can see that the panel on the left is the only one that really is considered negative, where there's really 0% staining. Yes.
Dr. Gandhi, I apologize. We aren't seeing your slides advance. Are you still pushing them forward?
Oh, you're not?
No, we're left on slide 12.
Do you see it now? I just tried to go back and forth.
Yep.
Okay, I'm sorry. I'm sorry about that. So this is on the very left. You see what's actually considered the only one that's actually considered 0% staining, where you see just no staining at all. But anything above that, depending on the trial, really could be considered positive staining. All three of the rightmost panels. There can be completely variable intensities of staining, as you see here. But really, what seems to have been most predictive, and I would say this is true probably for all of the therapies that are currently in development or approved, is really the proportion of tumor cells that express PD-L1, not the intensity per se. Atezolizumab, which is a PD-L1 inhibitor from Genentech, has a different antibody, different platform, and you can see here that they, again can see very similar intense staining. But what I think is really noticeable about this, and what was unique about the development of atezolizumab, is that they really tried from the very beginning of their phase 1 development to evaluate what are the differences that we should be looking at. And they looked not only at intensity and proportion of tumor cells expressing PD-L1, but they also looked at immune cells expressing PD-L1 in the microenvironment. When this was done early on in the development of Pembrolizumab as well, but was felt not to make a difference. But you can see here that in the early studies of atezolizumab, they defined multiple different populations, starting from the bottom of TC0 or tumor cell 0 and IC0, basically no expression whatsoever, and then really +1, +2, or +3 intensity of staining in either the tumor cell component or the immune cell component. And you can see that those populations overlap in the lower ranges, but once you get into high intensity staining, they almost seem to be mutually exclusive at the top of the page there, that tumors that express high levels of PD-L1 on the tumor cells don't seem to have a lot of expression on the immune cells, and vice versa as well. I also think what's notable about this slide is just to sort of point out that PD-L1 staining, no matter what the antibody you use, can be very geographic, that you can see areas of complete negativity next to areas of incredibly intense staining, as you can see on that top panel. And those are some of the issues actually that make it difficult to really understand how to best use PD-L1 as a biomarker. This study that is referred to on this slide, the Poplar study, was a randomized study versus docetaxel, a second-line chemotherapy used in non-small cell lung cancer. And in that original study, they actually saw that in this bottom group, the TC0 and IC0, that there really was no difference actually in the outcomes for patients who had either atezolizumab or chemotherapy. But there was a pretty dramatic difference as you went up in intensity, and that TC3, IC3 had basically the lowest hazard ratio or the greatest difference in using atezolizumab as compared with docetaxel. Now, between this study and a larger phase 3 study, they actually, although they kept the same assay and the same antibody, they switched vendors. And interestingly, in that study, they actually found that there was a benefit across the board. And ultimately, atezolizumab received approval for all patients in the second-line setting, regardless of PD-L1 expression. The same is true for Nivolumab, which was the first drug approved. Pembrolizumab is a little unique, and I will come back to that in a second. But it just sort of says to me that there's something, that we are not getting consistently right between all these different studies about looking at PD-L1. In fact, in that latter study, the percentage of patients who had TC0 and IC0 or completely negative staining was actually close to 45%, which is a much higher number that's been seen either in the earlier trials of atezolizumab or with any other agent, where that number is usually in the 20 to 25% range. So it suggests to me that not all of those negative patients were actually getting benefit, and some of them likely had some PD-L1 positivity. This is just a diagram to try to illustrate some of those issues in actually measuring PD-L1, even if you're using the same antibody. Both the primary tumor from the lung diagrammed here versus metastases may have different geographic patterns or different levels of staining. There can be changes in time of PD-L1 expression, since it's really a protein expression level that can be affected by multiple different things in the immune environment, including exposure to other therapies such as chemotherapy or targeted therapy. And there's just differences within the tumor. So the panel on the left sort of shows a more uniform stain, but there may be an area of the tumor where, just like we saw on the previous slide, you can actually sample the tumor and get a completely negative result, even though there are adjacent cells that are totally positive. Many times we also see, and this has actually frequently been described in melanoma, as on the right panel, that there are immune cells that surround the periphery of the tumor but are not able to penetrate into the tumor, and a lot of the PD-L1 staining seems to happen on that periphery at the interface of the tumor immune cell interaction. And again, you may or may not really sample that when you're actually trying to test for PD-L1 in routine biopsies or resections from patients. For lung cancer, unlike some settings of melanoma, biopsies have always been challenging, which is one of the challenges in actually developing treatments, because it's hard to get tissue to actually really evaluate what treatments are doing at that level. And biopsies are usually small, and they're usually limited. So they're invasive to actually enter somebody's lungs. Sometimes we can get additional tissue from elsewhere, but often it's actually a really limiting factor. But as I told you, with Pembrolizumab, they really defined a cutoff of 50%, where they thought they really did see benefits that differentiated outcomes for a subgroup of patients versus others. And when they came to do a first-line trial versus chemotherapy, so that initial chemotherapy that I showed you in the beginning slides of a platinum-based regimen, they did two trials, actually. One trial is still ongoing, which is comparing Pembrolizumab compared to first-line chemotherapy for patients of all levels of PD-L1 expression. And one was this trial, which actually only allowed patients who had high levels of PD-L1 expression into the trial. So only those patients were enrolled in this trial. And in this setting, you can see that there's a dramatic difference between Pembrolizumab and chemotherapy that just widens over time, with a large proportion of patients getting benefit and sustained benefit. This is actually progression-free survival. The next slide shows you the overall survival, which again, although this is still early data, this trial is actually still going on and there's still ongoing follow-up. So that's why you see both lines flatten out a little bit because of censoring. You can see this widening gap between Pembrolizumab and chemotherapy, which will likely continue to widen over time, with really the median overall survival not yet reached in these patients who were originally treated on this trial. This trial, which was reported in the fall of 2016, resulted in immediate FDA approval of Pembrolizumab for first-line treatment of non-small cell lung cancer, and it has really changed the way we treat lung cancer patients and also how we evaluate and generate data on their tissue during diagnosis to be able to make treatment plans. But I think I already have sort of told you that despite this now clinical use of PD-L1 as a biomarker of response and as a differentiating factor for treatment, there's a lot of skepticism in the field that that is the best biomarker to use, given all of the issues, both antibody-based issues, as well as sampling issues, as well as the dynamic change issues. And, you know, to some level, we don't really know what high levels of PD-L1 expression really means on immune compartments versus tumor cells. We do know from a large body of work to evaluate somatic mutation frequencies in different tumor types that the tumor types that have seen the most benefit from these types of drugs, PD-1 inhibitors and PD-L1 inhibitors, all have something in common. They all are at the end of this spectrum of tumors on the right with the highest mutational burdens. So the first approvals for PD-1 therapy came in melanoma. The seconds came in lung cancer, the third in bladder cancer, which are all, as you see here, the tumor types with the highest overall mutational burden, which makes some sense at some level, if there's a lot of mutational change in the tumor cells at the DNA level, we would expect a lot of potential neoantigens or altered antigens to be expressed, rendering the tumor more immunogenic or recognizable by the immune system. And in fact, this study, which was done actually on the basis of the Pembrolizumab initial phase 1 study, took those tumors from patients treated in that study and looked at overall mutational burden and could clearly differentiate that patients with high levels of mutations did much better and had much better outcomes than people with low level of an overall mutational burden. Now, when you use the specific gene sequences to actually predict what would be able to form neoantigens, I'm just going to forward for a second, we saw the same. I'm sorry, I think this slide came out differently. But basically, we saw the same thing, that neoantigen burden versus overall mutational burden looked basically exactly the same, that they both had a great predictive value for differentiating between response. For lung cancer, since we know that smoking is often has a linear relationship with mutational burden, that the longer people smoke for, the heavier smokers tend to have a greater accumulation of overall mutations. And if you look at what is considered a molecular smoking signature, which are a high rate of transversions, you also see that same differentiation. What we know also just from looking at clinical parameters in these studies is that patients who are smokers overall have a higher chance of getting benefit from these drugs than patients who are non-smokers. It doesn't seem to be as simple as just trying to look at smoking, though. In and of itself, it doesn't seem to have as much predictive value as either neoantigen burden, mutational burden, or PD-L1. And sorry, here's that neoantigen burden slide, which shows you that, again, you see this basically clear differentiation between patients with predicted high neoantigen burden versus those with low in terms of outcome to therapy. But the question still remains, are any of these things, the smoking-related signature, smoking itself, neoantigen burden, or mutational burden, actually better predictive markers than PD-L1? Certainly we know that PD-L1, which is an immunohistochemistry test that can be done in any pathology lab around the country, is a much simpler, much faster way to evaluate potential benefit than to try to actually get large-scale sequencing data and estimate overall mutational burden or neoantigen burden. The other concern is that, even if these things are potential surrogates for neoantigen, especially in melanoma where this work has been done in more detail and for longer, predicted neoantigens do not always translate into actual neoantigens, meaning that when you actually try to isolate T cells from the peripheral blood and actually stimulate them with predicted neoantigens, they do not always elicit T cell responses. A subset of them do, but it's not clear. There's not a clear way to know which subset that would be, and that might be variable from individual to an individual. That might be variable depending on what prior therapy that individual has received. That might be different depending on the disease or specific tumor type. So it can be really challenging to actually validate what's a true neoantigen, what is likely to be a really good predictive marker and a more specific predictive marker. The other problem is that neoantigen profile, just like PD-L1, can be dynamic. We know that elimination of individual neoantigens may be a mechanism of acquired resistance to PD-1 targeted therapy, but we also know that exposure to chemotherapy can actually change neoantigen profile, or exposure to other therapies may do so as well. So these are still active areas of evaluation that I think we still don't know the answers to. Beyond also what we can predict, even if we can predict accurately what might be a neoantigen, what might be a more specific biomarker, we know that there are many secondary mechanisms to impair T cell responses to neoantigens that might be pre-existing, but we don't have good ways to, again, evaluate those a priori prior to treatment. But those could be really the presence of myeloid-derived suppressor cells. Those are these green cells that you see, which actually can suppress the immune response. But it also can be sort of the fact that you might eliminate those cells that actually bear neoantigens and really select for a population that doesn't actually express a lot of neoantigens, that might be sort of non-immunogenic, those darker blue cells. We know that other kinds of cells can affect the overall response, including the T regulatory cells in the microenvironment, but also many other factors, including IDO in the environment, TGF-beta, other cytokines, many other things. So at the end of the day, even if you think you can predict what neoantigens might be there to elicit a response, there can be many factors that could impair or impede that response from happening. As I think I mentioned to you earlier, that in the development of Atezolizumab, Genentech, I think, was quite unique in really trying to look at multiple different factors as predictive biomarkers in addition to PD-L1. So in addition to looking at the immune cell compartment and the tumor cell compartment, they also looked at PD-L2 expression. They looked at a signature that they defined in their phase 1 study of a T effector cell signature or a gamma interferon signature as potential predictive markers. And what you can see in this forest plot at the top is that if you look at each one of these markers as a predictor value, they all show some predictive value, meaning there's a hazard ratio or an improved outcome with Atezolizumab compared to docetaxel, which is on the right. But none of them really look different from each other. And in fact, really they all look pretty similar to PD-L1 in and of itself. And those hazard ratios by numbers are numerically really no different than just looking at PD-L1. So we can put a lot more work into it, but at the end of the day, it's not clear that you get more information for doing that. So where we are now with these single-agent approvals is certainly in a better place than we were with chemotherapy. If chemotherapy is this line on the left, the dark blue line, and the shift is for that subset of patients who can get targeted therapies and do better for some time. The red line is really this subgroup who's getting benefit from immunotherapies. And there are really long-term survivors, and we really are in a very different place than we were. But it's still a subgroup at the end of the day. So I think it's important to remember that we want to do better for all patients. And if we know that there are ways to stimulate an immune response, and we know some of the mechanisms that can impair that, we theoretically should be able to overcome those and make non-inflamed or non-immunogenic tumors more immunogenic and more recognizable by the immune system. And that's where a lot of the current efforts are moving towards. So most clinical trials, I would say, of any type in lung cancer now are really focused on immunotherapy combinations, and the potential combination partners are many. So those could include chemotherapy and radiation, standard treatments in lung cancer and in many tumor types, because what we know about chemotherapy and radiation is that they theoretically should be immunogenizing. Chemotherapy is immunosuppressive, meaning that it actually should deplete myeloid-derived suppressor cells, but it also kills tumor cells and releases cytokines and releases potential neoantigens. So theoretically should make a tumor more immunogenic. And the same is true for radiation, which can cause a lot of local inflammation around the area of radiation. So that's an active area of research, and I'll come to that in a minute because that's already made some impact in lung cancer. But many of the ongoing studies are actually looking at oncolytic viruses, which has been an active area of research in the past, again, and ongoing, but maybe with additional checkpoint blockade. And checkpoint blockade really refers to, de-repressing or de-suppressing the T-cell response, either by PD-1 inhibition or by CTLA-4 inhibition or both. Maybe that will actually allow the oncolytic virus kind of response to be amplified. Similarly, vaccines, which have shown really minimal activity on their own, one of the reasons that might be is that there's local suppression at the tumor immune microenvironment that's preventing a significant immune response where you actually need it. So measuring T-cell responses in the blood after vaccine therapy may not be a good surrogate for what's really happening at the tumor level. So checkpoint blockade in combination with vaccines is an active area of research currently, as is checkpoint blockade with things like HDAC inhibitors or other agents that could theoretically turn on transcription and lead to improved neoantigen production. There are many immune combinations being evaluated as well, and I'll talk about one of these, but there are really hundreds of those going on with many other agents that we know act in the immune microenvironment, including IDO, 41BB or CD137, OX40, GITR, TIM3, LAG3, and so forth. I bring up this study because this is a study which has been completed and actually very recently, literally within the last week, just led to a new drug approval. But this is a randomized phase 2 study, so a pretty small study of only 120 patients, about 60 in each arm, who were treated with chemotherapy with or without pembrolizumab. So randomized study of standard therapy, and half got the addition of pembrolizumab as well. And you can see here that overall patients had a higher response rate when they were given combination therapy as compared to chemotherapy alone. And it's hard to tell whether PD-L1 was really a significant factor here. Certainly the high-level group had a high response rate of 80%, but the next highest group, 1 to 49%, had an actually lower response rate. So I think it's difficult to make much of these because the numbers are really quite small. But this higher response rate overall did lead to an improved progression-free survival, a hazard ratio of 0.53 for progression when you combine chemotherapy with pembrolizumab. And even though we see this difference, unlike with the pembrolizumab instead of chemotherapy study where we saw that translate into an overall survival difference, we did not see that translate into an overall survival difference here, although this measurement is still quite early on, and these results are due to be updated at a national meeting next month. Still, this certainly may be due to the fact that now many of these agents are approved in the second line, and there are patients who crossed over to receive the drug later and may have had just the equivalent benefit. But regardless of whether or not this will translate into really long-term differences in outcome, the FDA actually approved this combination through accelerated approval mechanism just last week. That's generated a lot of controversy in and of itself because many people in the field feel that there really isn't enough data to know who's actually benefiting from this therapy and who's not, and also what's really going to be a predictive biomarker to differentiate who's going to likely benefit. Although there's a different spectrum of side effects with pembrolizumab, which we haven't talked a lot about here, and chemotherapy, there can be additive side effects of combining all three drugs, so it's not without concern to do that. And this slide just shows you some of the many combinations that are going on in looking at immunotherapy combinations, both by combining PD-L1 or PD-1 inhibitors with other activating signals for T-cell stimulation on the left, but also with other inhibitory signals to enhance that effect of blocking PD-1-PD-L1 interaction on the right. And the one that has been most evaluated and has led to changes in treatment in melanoma, at least, is a combination of PD-1 inhibition with CTLA-4 inhibition. So in melanoma, where these studies have been going on for longer, it was the first drug, area I think I told you where Ipilimumab was approved. It was also the first area where Nivolumab was approved. It's also the area where they first evaluated combination treatment and actually demonstrated in large-scale studies, multiple large-scale studies, that combination therapy with Nivolumab and Ipilimumab is superior to monotherapy. And I think this study was meaningful in two ways. One, it showed that there was an overall improvement, that's the top panel here, with combination therapy compared to either monotherapy. But if you look at the middle panel, which are the PD-L1 positive patients, there was really no difference in these top two curves of combination therapy or Nivolumab monotherapy. So the greatest difference, as you see in the bottom panel, is in those who are PD-L1 negative. And this is really important for two reasons. One is that we don't want to give patients more therapies and more potential toxicities if we don't have to. But we also want to be able to give patients benefit who are unlikely to get benefit or less likely to get benefit with anti-PD-1 or PD-L1 therapy on its own. So this really sort of defines, I think, that there can be now broad and immune activity in multiple subgroups, that maybe you could use monotherapy for patients with high PD-L1, maybe you could use combination therapy for those with low PD-L1. I think at the present time, from a lot of my colleagues, there's still a tendency to use combination therapy because of the difficulty in defining who's high PD-L1 and low PD-L1, but hopefully that will improve over time. In studying this combination in lung cancer, it was, and I'm sorry, I'm just going to go back for 1 minute to sort of point out that there was a keen awareness that the difficulty with this combination is that toxicity is also greater with the combination. So the rate of what we call grade 3 or 4 or severe toxicities with the combination in melanoma was really upwards of 50%, meaning many patients were having really severe and difficult toxicities to deal with, even though they might get long-term benefit. One of the differences in the approach in development in lung cancer from the beginning was to actually look at different potential combinations. In lung cancer, ipilimumab monotherapy really has no activity. So if we were going to try to get synergy, it might just require some initial synergy without continued dosing as frequently as it is in melanoma. So in melanoma, ipilimumab is given much more frequently on the order in the combination of every 3 weeks and on its own every 4 weeks. In this study here in lung cancer, which was only done in the first-line setting, meaning patients who had never had chemotherapy, ipilimumab was given either in some cohorts, either at a rate of only once every 6 weeks or once every 12 weeks on the left. And what you can see is that in doing that, when you look in comparison to nivolumab monotherapy on the right, is that the overall rate of toxicities when you give ipilimumab less frequently is not that much greater than nivolumab alone. So that might be one way of mitigating the increased toxicity with combinations, is to change the dosing schedule and dosing combination. One thing that I think what we're still trying to interpret from this study is that unlike melanoma, where we saw that there was benefit overall, that the people who got the greatest benefit were probably those who were PD-L1 negative, when you look at this study, which is different because it's much, much smaller numbers. This is a phase 1 study, so there's really very small numbers of patients in each of these subgroups. But you can see that the response rate for either nivolumab or the combination, red or blue, goes up with increasing PD-L1 expression, with very high response rates on the very rightmost column in those who had the highest level of PD-L1 expression. It's a very small number of patients, only 13 patients, but the response rate was over 90%. So we're certainly very encouraged that this may be an even better therapy than monotherapy, but I think it still needs to be defined for whom and where that benefit will have the greatest effectiveness. Even though we're seeing that PD-L1 dependence, in this same study, we also saw that some groups that really did not get benefit from nivolumab monotherapy, such as patients who were non-smokers, which you see on the right panel, on the left panel, in the gray is nivolumab alone, blue with the combination, that the combination had much greater efficacy in patients who are non-smokers, that is greater than the overall 20% reported response rate with nivolumab alone in all patients. And there's still a greater response rate in smokers as well. In addition, when you look at subsets of patients who also did not seem to get as much benefit from PD-1 therapy, such as EGFR mutation patients, these are patients who have an alteration in the EGFR oncogene that really serves as a driver of growth and therefore is commonly associated with a low mutational burden because it's really one of the few things that's really driving the growth of the cancer. There were very small numbers of patients in the earlier trials, but overall they seem to have a much lower chance of benefit. You can see here in this panel on the left that only 7% responded to nivolumab as a monotherapy. Only 14% responded as a monotherapy. The numbers, again, are very small, 8 and 7. But much higher percentage overall, again, very small numbers, responded to combination therapy. So there may be a broadening of activity with using this combination in lung cancer as well. However, I think it's still an open question because this is a different combination of drugs, but very similar in terms of mechanism of action. Durvalumab is a PD-L1 inhibitor, so similar to Atezolizumab, and similar to avelumab. But overall, all of those therapies, both PD-L1 and PD-1, as monotherapies have pretty similar response rates. This combination with tremelimumab, which is made by the same company that makes durvalumab, was done, again, to evaluate the combination of a PD-L1 inhibitor with a CTLA-4 inhibitor. Tremelimumab is a CTLA-4 inhibitor. But here, unlike in the nivolumab ipilimumab study, we see almost a lesser effect in patients as the PD-L1 expression goes up. Now, you have to keep in mind the PD-L1 expression is being defined differently here because it's a different antibody, it's a different company, different drugs. But the highest level of response seems to be in those who have negative PD-L1 expression or low levels of PD-L1 expression. Another difference, however, in this study, which is still something we don't quite understand, is that in this study, all patients were previously treated with chemotherapy. In the prior study, they were all treatment naive, meaning they had not had any prior treatment, and that really may have an impact as well that I think we still need to sort out. So I think we're left in a good situation where we have many active drugs, that we have many potential combinations that can hopefully improve upon the activity we've seen so far, but we're really still trying to define the best way to identify the patients most likely to respond. The fundamental issue, I think, is that immune biomarkers are dynamic. These are not fixed genetic changes. They're really things that are affected by the environment around them and can be affected by prior therapy exposure, subsequent therapy exposure, which might have to do with the impressive effect of Pembrolizumab in the first line, and many other features about a patient's experience that we still have yet to identify. There's limited data from looking at targeted therapies, chemotherapy, or radiation, but there is some preclinical data suggesting that there can be upregulation of PD-L1 and increased mutational burden with exposure to these things. That's a lot of why some of these combinations were initiated. I think we still have to better define that, because certainly from the first-line trial of Pembrolizumab, we saw that the patients who really have had the best benefit, long-term benefit, and survival benefit are those that never had any chemotherapy, radiation, or anything else. So I think a lot of active work is going on in trying to define how other therapies impact the immune microenvironment. And that's an area that I focus on, is trying to look at actually paired biopsies to try to understand how patients who've been exposed to chemotherapy, radiation therapy, or targeted therapy might alter their immune microenvironment to make them either less or more likely to respond to subsequent immunotherapy. I think that's a really critical area of research, because it's very likely that most patients will be exposed to these other therapies. These tools for treating cancer are not going to go away. We haven't seen that all patients are getting cured with PD-1 inhibition as of yet. So this is just from actually a review by Tony Ribas, but I think is a very, very good illustration of where we are in the field, where we would like to be. We've seen that there's a subset of patients that are getting long-term survival with PD-1 inhibition, but we'd like that tail of the curve to be raised for a large group of patients, so that the majority of patients are actually living and are long-term survivors of metastatic cancer with effective therapies. And we hope that we can get there with immune checkpoint blockades in combinations, or potentially with actually the appropriate sequencing of therapy. And these are still things that I think we have to sort out. They're difficult trials to do because of how quickly the field is moving, how quickly drugs are getting approved, and how many patients have already been exposed to these drugs. So it's a challenging area to work in, but also very exciting. One of the other areas, obviously, if we can do this well with metastatic disease, we obviously would want to move all of these things earlier in the development of cancer. If we can actually make long-term benefit in the metastatic setting, could we increase the rate of cure from surgery in the earlier setting? There are many ongoing studies of these agents either after surgery in the adjuvant setting, postoperative setting, as well as prior to surgery in the neoadjuvant setting, and even some studies that are being planned for looking at whether we could treat intrapulmonary nodules or dysplasia, things that are not even cancer yet, with immunotherapies to help prevent them from becoming that way. The study of durvalumab in the adjuvant setting actually had a press release again just last week that suggested that there was an improved progression-free survival compared to those who just had standard monitoring. So that may be an area where there's again going to be a standard of care change very quickly because we're already seeing benefit. It remains to be seen, though, I think, whether that will truly translate into survival benefit. Even with some of the vaccines that were tested in that kind of setting, we saw some progression-free survival benefit that did not translate into survival benefit. There is a lot of excitement, however, about neoadjuvant studies where we're actually able to look at paired tissue biopsies and see what's happened to the tumor from the immunotherapy. This is actually from a phase 1 study done at Johns Hopkins from Patrick Ford, where patients received nivolumab prior to getting resection of their early-stage lung cancer. And you can see in the red that there were close to 40% of patients had what we would call a major pathologic response, or killing of the tumor such that there was less than 10% residual viable tumor left at the time of surgery. We still don't know whether this is going to translate into long-term survival, but what we know about chemotherapy in this setting suggests that that is likely to be the case. So there are now ongoing randomized trials to evaluate nivolumab versus chemotherapy as neoadjuvant therapy, as well as combination of nivolumab plus ipilimumab as neoadjuvant therapy. And from, again, this phase 1 study, one of the things I think that suggests that this may be an effective way of doing things is in this pretreatment specimen on the left, you can see that there's not a lot of immune infiltrate. There's not a lot of CD8 or green cells in that environment, not even a lot of PD-L1 positive cells, which are white. But post-treatment, there's a really massive immune infiltrate that's come into the specimen as a result of exposure to the therapy. So while we tend to think that you need both immune cells as well as neoantigens to generate an immune response, there's certainly the thought that, you can stimulate that kind of effect with exposure to these therapies, and maybe the earlier we do it, the more we'll see it. And lastly, I just wanted to sort of touch on the fact that PD-1 inhibition, checkpoint inhibition is only one way of stimulating tumor immunity. In the world of hematologic malignancies, there has been a parallel and really exciting development of sustained long-term survivors or long-term remissions in lymphocytic leukemias and lymphomas with the use of CAR T cells, which are chimeric antigen receptor modified T cells that are basically taken out of the patient's body, modified to be responsive to neoantigen and put back into the body and generate a significant immune response. And this has really resulted in quite dramatic effects that are, again, being studied in a much larger way, and including in solid tumors now. These types of therapies, however, are not without their problems as well, in addition to potential toxicities, which I might say are even potentially greater than in the world of checkpoint inhibition, because they lead to really inpatient hospitalizations and often very dramatic cytokine release syndromes. There have actually been a few patient deaths from cerebral edema, including one that was reported about a week ago. The cost of doing this type of therapy is very, very high because it has to be personalized and individualized for every individual. All of these therapies are expensive, don't get me wrong. The costs for PD-1 inhibitors are astronomical and really new and unheard of in the world of oncology. But CAR T cells go one step further. So those are things that we need to actually be mindful of in further development as well. It's not yet clear where the future is going to be for all of these therapies. I would just say in conclusion that it's really an exploding area in lung cancer and all cancers, with many, many combination trials underway or planned. But I think we're going to have to be patient, because the difference will not be in seeing different responses or even necessarily progression-free survival. What we really want to see are that they translate into long-term overall survival differences. And it might be harder to see that for a while with the combinations, because we already see some responses early on with monotherapy. Biomarker evaluation is also really exploding, with many, many things being looked at now in clinical trials rather than just PD-L1. But it is going to be a challenge, I think, to sort out the best clinical path forward. What is the most utilitarian type of biomarker that can be used in a standard clinical setting in a rapid way to enable real-time care for real people? And I will stop there and answer questions.
Thank you, Dr. Gandhi. Our audience has busily been asking questions throughout your presentation, so let's get started with the Q&A. How do you see precision medicine informing immunotherapy choice progressing over time?
I think that's a very important question. Precision medicine used to mean actually looking for oncogenic drivers or fixed genetic mutations that drive the growth of the cancer so that we could develop targeted therapies. And really, we could define a subset of things, a finite number of those. Precision medicine going forward is actually really about defining individual mutational predictors of immunotherapy response. So that means both looking at the DNA level, looking at mutational burden, predicted neoantigens, as well as oncogenic drivers. There's also a concept, though, of functional precision medicine, which we are getting closer to. There are several groups actually trying to look in real time in short-term in vitro organoid cultures to try to evaluate actually what changes in the immune microenvironment with exposure to cytokines or exposure to therapies that might actually define how a patient is likely to respond. Because certainly, especially in terms of immunotherapy, looking at the DNA or predicted neoantigens is really only one part of the picture. There's so much more going on at the protein level in the microenvironment that we may not be able to detect by just looking at the DNA. So I think precision medicine is going to get a different definition going forward, and it is going to have to become more functional to really be useful.
Thank you. This question just came in. You mentioned that chemotherapy or radiotherapy might affect how downstream immunotherapy functions. Does surgery also have this effect, and does it impact the local circulation of the immunotherapy therapeutic?
So in terms of impacting the local circulation, it should not. But I think that in terms of inflammatory effects, it absolutely can. And the same thought is true for surgery and as for radiation, that basically we can generate an increased immune response potentially from the inflammation that occurs after surgery or exposure to radiation. I think that still remains to be proven, however, because again, what we see from these first-line trials where patients have not been exposed to any prior therapy is that's where we're seeing the greatest difference. In fact, there actually have been 2 large-scale first-line trials done. I only talked about the Pembrolizumab trial because it was positive. There was a similar study of looking at Nivolumab in combination, instead of chemotherapy. So patients with all levels of PD-L1 were randomized to either Nivolumab or standard first-line chemotherapy, and that was a negative trial. And we're still actually trying to understand why it was a negative trial when the Pembrolizumab study was positive. There were some imbalances in the arms, but one of the differences overall is that there was, for some reason, a high rate of patients who had received prior radiotherapy, actually almost 40%. I don't know why that is exactly, whether these were patients who had received some local radiotherapy, small doses. We don't really know any of the details, but it makes you wonder whether we can really assume that radiation-related inflammation is truly a positive thing when it comes to subsequent immunotherapy. I don't think we know that yet.
Thank you. This is a follow-on question. What's your opinion on how immunotherapy works in treatment-naive patients versus treatment-experienced patients? Do you have any clue on the origin of that difference?
I think it's a very good question. I don't think anybody knows for sure, but I think there is some thought that it can potentially affect the outcomes through subsequent treatment. So even patients who are long-term survivors, many of them end up having some progression of areas of disease during the course of their treatment or following their treatment. So it's not that everybody sort of just gets cured and everything disappears. For most patients, what we're seeing are partial responses. There's tumor left. And for most patients, it progresses over time. And so for most of those patients, they go on to receive subsequent therapy, usually with chemotherapy. So there is some thought that that immunotherapy could actually prime a chemotherapy response. And certainly in the second-line setting and where we've had experience of patients who've gone through phase 1 trials and then moved on to other therapy, that they're living longer than we thought they would otherwise with exposure to standard third-line or fourth-line therapy, and that there may have been some kind of an effect of the immunotherapy on the subsequent response to the chemotherapy. So I don't think that's just pure speculation. I don't think we know that as yet.
Thank you. That was very interesting. Are you able to comment at all on the difference between the mechanism of action of Pembrolizumab versus Nivolumab?
So as far as we know, there's really no difference in the mechanism of action, actually. They're very similar antibodies. Pembrolizumab is an IgG4, and Nivolumab, I believe, is an IgG2, but they're otherwise very similar. They're very similar in their mechanism of action, and they're very similar in terms of their clinical data and how effectively they work or not work.
Great, thank you. Do patients or their tumors develop resistance to PD-1, PD-L1 immunotherapies?
Yes, most patients develop resistance. So only, as I showed you, only about 20% of patients end up being long-term survivors. Most people develop resistance either from the get-go or over time. And we're still trying to define some of those mechanisms of resistance. I think I mentioned to you that we know that the loss of neoantigens may be one mechanism of resistance. There's been some suggestion in lung cancer studies actually that TIM3 upregulation can be a mechanism of resistance, LAG3 upregulation, or other kinds of immune factors in the microenvironment. But I think we're still really at the very starting point of defining those mechanisms.
Thank you. You mentioned that accessing diagnostic tissue is difficult in lung cancer. Is a liquid biopsy of lung lavage a diagnostic option?
So that is an option, but it's a very limited option. So while we use those types of things to look at DNA from tumor cells that might be in lavage or in the circulation, and we could get information on the tumor mutational burden, I suppose, from that. What you can't get any information about is the entire immune microenvironment. So without a pathologic tissue specimen, you cannot see actually where there are cells expressing high levels of PD-L1, where there are T regulatory cells in the environment, where there might be things on the periphery of the tumor versus in the tumor, how much of an immune infiltrate, and what are the particulars of that. You can't get any of that from those types of specimens.
Got it. Is the resistance that you see with immunotherapies related to anti-drug antibodies?
It's a good question. So there has been some of that reported, but they're very isolated circumstances, and it's not clear that that's really the mechanism of resistance. That might be a mechanism of toxicity, but not clearly a mechanism of resistance.
Understood. We have 2 questions left. When conducting immunohistochemistry on tissue biopsies after use of therapeutic antibodies, is there a risk of masked epitopes and not finding what you're looking for?
I'm sorry, I didn't quite catch that. What was the full question?
When conducting immunohistochemistry on tissue biopsies after the use of a therapeutic antibody, is there a risk that you're going to have masked the epitopes that you're looking for?
Yes, there definitely can be. So you mean after the use of anti-PD-L1 antibody, for instance?
Yes.
Yes, there definitely can be. I mean, I think there are mechanisms to get around that, to unmask antibodies. And also, that it's not that you're saturating necessarily all the receptors all the time. So I don't think we know how much of an issue that is, really. I mean, we certainly see, for instance, when you look at post-treatment biopsies, we see frequently that PD-L1 is upregulated. So we're seeing a lot of that expression. And we see actually upregulation of PD-1 as well. We see increased exhausted tumor cells, exhausted T cells. So it's not that we're necessarily masking the epitopes, because to be able to see that, you can't have.
And our final question, are there any other receptor ligand pairs that could work in a similar way for other types of lung cancer?
I'm not sure exactly what's the, intent behind that question. So other types of lung cancer are also potentially responsive to PD-1, PD-L1 interactions. And we've seen that for small cell lung cancer, although actually we know that PD-L1 expression in tumor cells themselves may be a lot lower, although there are more frequent tumor-infiltrating macrophages that can have high levels of expression. In the studies to date in small cell lung cancer, we've seen more activity with combination therapy with CTLA-4 antibodies and PD-L1 antibodies, or PD-1 antibodies, and then with monotherapy. But it's still a much newer area of study than with non-small cell lung cancer. We know that mesothelioma, which is a thoracic cancer of the lining of the lung, can have high levels of PD-L1 expression in tumor cells. And we have seen responses in patients with mesothelioma. It remains to be defined, I guess, what that true rate of response is, whether it's as good as lung cancer, and what the impact of combination therapy might be.
Thank you. Unfortunately, that's all we have for today. If you have any further questions, please consider reaching out to Dr. Gandhi. Her email address is shown on the screen. No, it's not. Hold on one second. Let me make sure it gets there for you. There we go. As a reminder, the webinar will be archived on the scientist website, and you'll receive an email notifying you when the on-demand webinar is available. I'd like to thank everyone who took the time to join us today, and particularly those who shared your questions and comments. On behalf of the scientists, I'd also like to thank Dr. Gandhi, as well as our webinar sponsor, Beckman Coulter Life Sciences. Thank you, everyone, and goodbye.